key to any successful translational therapy is balance. It's finding a balance between a treatment that's effective but doesn't lead to any unwanted side effects. My thesis research is focused on finding balance in treating a rare genetic skin disorder known as recessive dystrophic epidermolysis bullosa. This disorder is caused by mutations in a crucial gene known as type 7 collagen. When these mutations occur, patients suffer from a number of complications, including chronic skin blistering, chronic infections, fusion of the fingers and toes, as well as a lethal form of skin cancer. And while there's currently no cure for this disease, our lab has made great strides in the use of bone marrow transplant to treat this. And as you can see, before transplant, patients suffer from these chronic blistering throughout their body. But shortly after transplant, we see a dramatic improvement in the overall skin integrity and an improvement of the symptoms as well. But we didn't want to stop there. In order to make the therapy as effective as possible, we wanted to increase the amount of collagen 7 that the transplant made, which meant understanding how collagen 7 is regulated. And so before I started in the lab, not a lot was known how collagen 7 was regulated at the RNA level, specifically through a particular type of regulation by a small class of RNA molecules called microRNAs. Now, these microRNAs are very small, but they play a very important role in regulating large groups of genes. And I determined that a particular microRNA known as miR29 regulates collagen 7 very robustly in a negative fashion. And this is very similar to a balancer scale, where when we see low levels of miR29, collagen expression is high, and vice versa. And we wanted to exploit this, but we ran into a conundrum. You see, miR29 also regulates a lot of the genes that are associated with fibrosis, leading to the fusion of fingers and toes, and skin cancer. And so we didn't want to cause any unwanted side effects with this therapy. And so instead, we had to figure out an alternative approach. And that meant designing a new therapy or a new type of molecule, which we call a target protector. This target protector allows us to remove collagen 7 from the scale. It specifically blocks miR29's inhibitory and interaction with collagen 7 while leaving all of the other genes untouched. And so miR29 can still keep those down. And so when we use these collagen 7 target protectors, we see an increase in collagen 7, but we're not promoting fibrosis and skin cancer. And we hope to use this for future therapies and finding a balanced way to treat recessive dystrophic epidermolysis bullosa. Thank you.